I am going to discuss today a very basic topic in biochemistry, which is inhibition of enzymes. So, coming to the definition, the reduction or complete stoppage of activity of an enzyme by the influence of another substance in the enzyme mixture or in the living cell is called enzyme inhibition. So, inhibition occurs either in laboratory practice where the enzyme is assayed as well as in the natural cases in the living cell. It is essential. And the chemical substance which causes the enzyme inhibition is called the enzyme inhibitor. And the enzyme inhibition is a major regulatory mechanism in living cells. For example, in metabolic pathways, uh, some of the enzymes are having this regulatory nature and they control the flux of the substances through this pathway. Enzyme inhibitors are commonly used as drugs, preservatives, and poisons. So many of the drugs used in modern medicine are actually enzyme inhibitors. And if some enzyme in our body is overactive and this causes the uh, disease, we have enzyme inhibitors used as drugs. And also food preservatives. They prevent the action of several enzymes in bacteria, etc., and act as preservatives. And poisons such as insecticides, rodenticides, fungicides, etc., act by binding to several enzymes and preventing them and act as poisons. So, coming to the classification of enzyme inhibition, they are broadly classified into reversible and irreversible types of inhibition. Reversible inhibition is classified into competitive, non-competitive, and uncompetitive types. Whereas irreversible inhibition is classified into substrate analogs, group-specific agents, as well as suicide inhibitors. So we shall see all of them one after the other. So coming first to reversible inhibition. In reversible inhibition, the inhibitor binds to the enzyme by non-covalent interactions such as hydrogen bonding, hydrophobic interactions, van der Waals interactions, salt bridge interactions, etc. No covalent bonds are not formed. So the enzyme inhibitor complex can dissociate very easily into the enzyme and inhibitor. So this term is called reversible inhibition because it can revert back into enzyme and inhibitor. In irreversible inhibition, covalent bonds are established between the enzyme and the inhibiting molecule. So once formed, the enzyme inhibitor complex does not dissociate, permanently destroying the enzyme. So it is called irreversible inhibition. It cannot be reversed because covalent bonds are very strong and they cannot be dissociated easily. Types of reversible inhibition. So coming to the different types of reversible inhibition, it is classified into three types. First one is competitive inhibition. Here, the inhibitor binds to the active site of the enzyme, preventing the binding of the substrate. The inhibitor and the substrate are mutually exclusive. Mutually exclusive meaning either the inhibitor will be binding or the substrate will be binding. Both of them cannot bind simultaneously with the active site or the enzyme. So that is called a mutually exclusive. And then inhibitor may be a non-metabolizable analog. So it is a substance which binds to the enzyme, but which cannot be metabolized or converted to product by the enzyme or derivative of the substrate. It may be looking similar to the substrate and will be binding to the enzyme, but it cannot get converted to product or product of the reaction. Or product of the enzymatic reaction will be similar to the substrate and it can act as an inhibitor or an alternative substrate of the enzyme. So the enzyme may be having different substrates, and in the presence of one or two more substrates, it can be acting as a inhibitor for the first one. In the presence of large amount of substrate, all the enzyme will be converted to enzyme substrate complex, and the effect of inhibitor will be negligible. So this in competitive inhibition, it is observed that if we add a large amount of substrate, the most of the enzyme will be in the enzyme substrate form. and the effect of the inhibitor will be very less. So this can be easily reversed by adding a large amount of substrate in the SA mixture. So this is the scheme of competitive inhibition. We can see on the left hand side enzyme plus substrate giving enzyme substrate complex and which gives the enzyme product, and this enzyme can bind with the inhibitor forming enzyme inhibitor complex, which is not converting into products. So substrate is competing with the inhibitor to form the complex. So the Vmax will be unaltered in the case of competitive inhibition. It will be the same. And Km will increase as a portion of the enzyme will be in the EI form, which has no affinity for the substrate. So the enzyme inhibitor complex once formed will not bind with the substrate. So the effective Km will be increasing. Vmax will not be altered because we can add more and more substrate, and this can almost cancel the effect of this inhibition. So we get the normal Vmax in the SA condition, but Km will be increasing. And this is the velocity substrate concentration and Lineweaver-Burke plot of competitive inhibition. On the left hand side, you can see the velocity substrate concentration plot, which shows that Vmax is the same and Km is increasing. And this is more clearly seen in the Lineweaver-Burke plot, where we can see the same 1 by Vmax and altered 1 by Km, minus 1 by Km. So this Lineweaver-Burke plot is very useful for determining the type of enzyme inhibition. At one glance, we can see the type of the inhibition, whether it's competitive, uncompetitive, non-competitive. Second type is non-competitive inhibition. In non-competitive inhibition, the substrate and the inhibitor bind reversibly, randomly, and independently at different sites of the surface. So, in the case of competitive inhibition, we saw that enzyme and the substrate and inhibitor are binding to the same site. They are competing for the active site. But here, there is no such thing. They are binding at a different sites, which are both are reversible. Then randomly means either the substrate can bind first or the inhibitor can bind first, and independently at different sites on the enzyme surface. And the resulting ESI complex is inactive. So the enzyme may first bind with the substrate forming ES complex and immediately bind with the inhibitor forming ESI complex. Alternatively, the enzyme can first bind with the inhibitor forming the EI complex, which then binds with the substrate forming ESI complex. And this ESI complex is inactive. It cannot form products. At any inhibitor concentration, a high substrate concentration cannot drive all the enzyme to the ES form. A portion of the enzyme will always remain as inactive in ESI form. So Vmax will decrease and Km will be constant as E and EI have equal affinity towards S. So Km is the affinity of the enzyme towards its substrate. That won't be affected because both free enzyme and enzyme inhibitor complex has the equal same affinity towards substrate. So that is not altered. But Vmax is decreased because some of the enzyme will always be present in the ESI form, which is inactive. So the amount of enzyme in the SA mixture will get reduced. So this is the scheme of non-competitive inhibition. It's a closed scheme. On the left hand side, we can see enzyme binding with the substrate forming ES complex, and it can bind with the inhibitor also forming EI complex. And both ES as well as EI complex can bind with this inhibitor or the substrate forming IES or ESI complex, which is inactive. So this is a scheme, a closed scheme of this non-competitive inhibition. So coming to the velocity substrate concentration plot of non-competitive inhibition, here we can see the Vmax is decreasing and Km is constant. And this is more obviously seen in the Lineweaver-Burke plot of non-competitive inhibition. We can see the minus 1 by Km is constant and 1 by Vmax is altered. So this is more suited for the determining type of inhibition. Now, thirdly, we shall see uncompetitive inhibition. This is the third type of reversible inhibition. In uncompetitive inhibition, the inhibitor binds to the enzyme substrate complex and not the free enzyme. So, this is an interesting case. If we mix the free enzyme and the inhibitor in molecule, it may not bind. Only if the substrate is present, enzyme will form enzyme substrate complex, and this complex will be binding with the inhibitor. Some structural change occurs in the enzyme substrate complex, which makes it possible for the inhibiting molecule to bind. Enzyme binds to the substrate forming ES, and this ES binds to I forming ESI complex, which is inactive. So enzyme is inactivated as ESI complex. At any concentration of the inhibitor, a high substrate concentration will not drive all the enzyme to the ES form. Some ESI form will be present, so the Vmax will be decreasing. And the Km also decreases due to the reason that the reaction between E and S increases due to Lee-Chatelier principle. So you might have studied about Lee-Chatelier principle in chemistry. That when a constraint is applied on a system at equilibrium, the system will change in such a manner so as to cancel the effect of this change. So, when we have the enzyme and substrate and the inhibitor, enzyme will be interacting with the substrate first, and then this ES complex will be uh, reacting with the inhibitor, forming ESI complex. This removes the enzyme from the reaction mixture, and more and more enzyme will be uh, reacting with the substrate to form ES complex in according to Lee-Chatelier principle. So the scheme will be decreasing. So this is the scheme of non-uncompetitive inhibition. On the left hand side, we can see enzyme plus a substrate giving ES, and that can react with the I giving ESI complex, which is inactive. ES will be converting to E and P. So this is the scheme of uncompetitive inhibition. So this is the Lineweaver plot of uncompetitive inhibition. A series of parallel lines are obtained. The lowest line is with no inhibitor, a native enzyme, and when we increase the concentration of the inhibitor, several parallel lines are obtained. So this is the velocity substrate plot of non-uncompetitive inhibition, and we can see the alteration in the Km as well as the Vmax. So Lineweaver plot is the most apt plot for the determining of the type of inhibition. So if you want to determine the type of inhibition, what you have to do is you have to run the assay with the different concentration of the substrate, same concentration of the enzyme and inhibitor, and you have to calculate the Lineweaver-Burke plot, draw the Lineweaver plot, and from that you can easily see the type of inhibition by comparing with the native enzyme. Okay, next type of inhibition is irreversible inhibition. Irreversible inhibition occurs when the inhibitor binds covalently to the enzyme and the complex form dissociates very slowly or does not dissociate at all. So you know that covalent bond is very strong. Once formed, it cannot be broken easily. And this irreversible inhibition causes the uh, formation of covalent bond between the enzyme and the inhibiting molecule. Three types are known generally. First one is group specific agents. They react with specific side chains on the amino acid of the enzyme. So you know that the enzyme is composed of amino acids, and it will have several side chains. And one of the side chains may be reacting with the inhibitor forming a covalent bond. And these are called group specific agents. So these are inhibiting compounds which react with the specific side chains on the enzyme with the covalent bond. So once formed, they won't dissociate, and the enzyme will be permanently uh, dis- uh, destroyed. So that's the term irreversible inhibition. It cannot be reversed easily. Second one is affinity labels. They are structurally similar to the substrate but bind to the enzyme with a covalent bond. So you know that enzymes are binding with the substrate by non-covalent interactions only. But here in affinity levels, these inhibiting molecules are structurally similar to the substrate, but they bind with the enzyme with the covalent bond. So once formed, the enzyme cannot bind the substrate. So it is permanently destroyed. Third one is suicide inhibitors. They bind to the enzyme like a substrate and is converted by the enzyme to another compound. This binds with the enzyme forming covalent bond, destroying it. So this is a very interesting case in which the sub-inhibiting molecule binds with the enzyme like a substrate, just like a substrate, maybe by non-covalent interactions. And this is converted to the enzyme to form another compound. So enzyme is acting upon the inhibitor just like a substrate and forming another compound. Now this second compound binds with the enzyme forming covalent bonds, destroying it. So this enzyme is literally committing suicide. It is binding the inhibitor, forming a second compound, and the second compound can bind with the enzyme covalently and permanently destroy it. So these are the different types of irreversible inhibition. So coming to some examples, group specific agents, diisopropyl phosphofluoride binds with the serine of the enzyme. So this compound, diisopropyl fluorophosphide, is binding with the serine of the this enzyme with the covalent bond, and it destroys the enzyme. So wherever serine is involved in the catalytic process or present in the active site, this can be acting as a irreversible inhibitor. So coming for affinity labels, bromoacetol phosphate, which is an analog of dihydroxyacetone phosphate that covalently modifies a glutamic residue in the active site of the enzyme, triose phosphate isomer. So this is involved in photosynthesis, etc. And this enzyme is inhibited permanently by bromoacetol phosphate. And suicide inhibitors. A common example is aspirin. It's a common painkiller. It is called acetyl salicylic acid and it inhibits the cyclooxygenase enzyme which is involved in the inflammatory process in our body. So aspirin acts as a substrate analog and enters the active site of the COX enzyme. And the enzyme attempts to process it, causing aspirin to acetylate a specific serine amino acid. This permanently blocks the active site, stopping the enzyme from synthesizing prostaglandins, which trigger pain and inflammation. So, aspirin is a suicide inhibitor for the enzyme cyclooxygenase. It first reacts with the this cyclooxygenase and gets destroyed. So, this is several examples of irreversible inhibition. So to recapitulate, enzyme inhibition also of two major types. One is reversible, in which only covalent bonds are not formed. Only the non-covalent interactions are established between the enzyme and the inhibitor, just like substrate. Whereas in the case of irreversible inhibition, covalent bonds are formed between the enzyme and the inhibitor, and this causes the permanent destruction of the enzyme. So thank you.